A very good morning to all of you. So today just continue our second part of the questions which are important for the Grand Viva for the UG students. Okay, first part already I shared on my channel. If you don't watch that, please go through that. Okay, so let's start the topic. In lipid chemistry we had done. Okay, now comes on the lipid metabolism. So what important things you must have to study for your Grand Viva and lipid metabolism is that of beta oxidation of fatty acid. In beta oxidation of fatty acid, you must read why beta oxidation is known as beta oxidation. Why the name beta is given to this oxidation. Okay. Second is the site. What is the site of the beta oxidation? Next important thing is that of energetics. That how much ATP is formed if there is an oxidation of your palmitic acid, and how much ATP is formed if there is a stearic acid, and how this beta oxidation is regulated. Okay, so these are the important things. Next comes of your that is odd chain fatty acid oxidation. You should know the proper step as well as the name of enzyme, that how the succinyl CoA is synthesized from the propionyl CoA. So all the steps of the synthesis along with the name of enzyme you should know. So important for your Viva. Next important is disorders of beta oxidation. You must know the name of all disorders which are associated with the beta oxidation. Okay. Next important is what is the role of peroxisome in beta oxidation. Okay. What is the significance of peroxisome related to the fatty acid oxidation, that you should know. Alright. Now next comes another important is that of cholesterol. Fatty acid synthesis is never asked in the Viva, so you can skip that. Okay. So next thing is that of your cholesterol. In cholesterol, the important thing first thing you should know the structure of cholesterol. How many carbon atoms are present in cholesterol? Answer is 27 carbon. Okay. So first thing you should know the structure. How many carbon atoms are present in cholesterol? At which position of cholesterol the hydroxyl group is attached? Okay, at what position of cholesterol ring the hydroxyl group is attached. Third important thing is what is the full form of CPPP? C-triple-P. What is the full form of C-triple-P? Okay, that you should know. So C-triple-P is, as you know, it is cyclopentanoperhydrophenanthrene ring, because the phenanthrene ring is there. Okay, so you should know the full form of CPPP, C-triple-P. Okay, now important thing is that as I told you before, that in every metabolism of all biochemistry, each and all metabolism, you should know the rate limiting enzymes. Okay, so as such you should know what is the rate limiting enzyme of your cholesterol biosynthesis. Another important thing is that what is the mechanism of action of statins to lowering the cholesterol. Why atorvastatin and rosuvastatin is prescribed to a patient of having a high cholesterol, and what is its mechanism of action. So you should know. Next important is that of degradation of cholesterol. What are the degraded product of cholesterol? That you should know. Next is that of what is name of what is primary bile acid, what is secondary bile acid, and what are the bile salts. Primary bile acid, secondary bile acid, and bile salts. You must know. Okay. Now next come on your ketone bodies. Very important topic, ketone bodies. Why? So first thing you should know the name of all three ketone bodies: acetone, acetoacetate, and beta hydroxylate. All three ketone bodies name you should know. Out of these three ketone bodies, which is a primary ketone body, which is secondary ketone body, and why primary is called primary and why secondary is called secondary? What is the reason behind that? Next important, out of these three ketone bodies, which of the following is a false ketone body and why? The reason you should know. Next is what is the precursor molecule from which the ketone body synthesis will take place? What is the precursor? Precursor is your acetyl CoA. Okay. Important thing regarding acetyl CoA is that you should know the fate of acetyl CoA in the body. That is, where in which cycle or what are the things that acetyl CoA act as a precursor. So you should know all, like ketone bodies, cholesterol, everything you should know where the acetyl CoA is precursor molecule. That is important question for your Viva. Okay. Next important thing is that of that why liver cannot utilize the ketone body itself. Although ketone body synthesis occur in liver, but when ketolysis will occur, then liver cannot utilize itself the ketone bodies. What is the reason behind that? That you should know. Next, you should know the ketosis. What is ketosis? It includes ketonemia and ketonuria. So, what is the value of when you will say the ketonemia? When you will say the ketonuria. Next, which test you will perform for ketone body detection? So, you will do Ruther's test. Then Ruther's test, you should know what is the step of Ruther's test, what is the principle of Ruther's test. Next important that out of 3 ketone bodies, which ketone body will not give Ruther's positive and why? So answer is that of beta hydroxybutyrate. Then if beta hydroxybutyrate cannot give Ruther's test, then what test you will perform in the lab to identify beta hydroxybutyrate? That answer you should know for your Viva. Okay. Next important DKA, diabetic ketoacidosis. As I told you before, you should know DKA. Okay. It is a medical emergency and how you will manage the patient of DKA and what clinical features that are presented in the patient of DKA. What is Kussmaul breathing? What is the reason of hyperventilation? That all things you should know in the DKA. Okay. Now next come on the chylomicrons. Chylomicrons important is that of you should know the thing which is bad cholesterol, which is good cholesterol, why bad cholesterol is called bad, why good cholesterol is called good. This thing. Next important thing, you should know the metabolism of your HDL. Okay. In HDL, important thing for the Viva, that is what is reverse transport of cholesterol. Okay. What is reverse transport of cholesterol? Another important thing, LCAT. What is LCAT? What is the full form of LCAT and what is the role of LCAT in HDL metabolism? You should know very well. Okay. What is difference between lipoprotein lipase and hormone sensitive lipase? Okay. And where this lipoprotein lipase will occur and where this hormone sensitive lipase will occur. That you should know. Another important thing is that of Frederickson classification. All the classes, how Frederickson classified the hypercholesterolemia. And out of all the classes, at least the thing you should know is in type 1 and type 2A. In Frederickson classification I am telling you. So in type 1 and type 2A, these two. So in type 1 you should know what is the defect in type 1, what is the defect in type 2A. Okay. Next important thing is where eruptive xanthoma will occur and where tendon xanthoma will occur. Okay. So this is about your Frederickson classification. Important thing is that of what is Tangier's disease. Okay. What do you understand by the Tangier's disease and how this disease will occur? So Tangier's disease is important for your Viva point of view. That's all about your chylomicrons. Now comes on your fatty liver. Fatty liver, what is the definition of fatty liver? At what diagnostic criteria you will say the patient is suffering from fatty liver? Because a normal amount of fat is accumulated in liver. Then you cannot say all patient is suffering from fatty liver. Then at what stage, that is at what amount of fat should be accumulated in hepatocyte, that you will say patient is suffering from fatty liver. That diagnostic criteria you should know for the fatty liver. Another important thing is that what are the types of fatty liver. So it's a alcoholic and non-alcoholic fatty liver disease. Okay. Next important thing is that what are the lipotrophic factors. Okay. And what is the role of lipotrophic factor in developing of fatty liver. That is important. Next important thing is that of your metabolic syndrome. What is metabolic syndrome? What is the another name of metabolic syndrome? Okay, because metabolic syndrome has two other more names. So what is metabolic syndrome? What is the other name of metabolic syndrome? And what is the diagnostic criteria of metabolic syndrome? How you will say the patient is suffering from, how you will diagnose the patient is suffering from metabolic syndrome? Because out of the 5, at least 3 should be present in the patient. Okay, while you will study, you will just read that out of 5 criteria, if when patient is having out of 5, 3, then we can say patient is suffering from the metabolic syndrome. Okay. Next important is that of in atherosclerosis. Now comes on atherosclerosis. So atherosclerosis, at least in brief, you should know how the atheromatous plaque is formed and what is the treatment and risk factor of atherosclerosis for Viva point of view. So how briefly, you will not describe all the mechanism, at least the steps. Okay, what are the steps of atheromatous plaque formation? What are the risk factor of your atherosclerosis, and what are the management? How you will manage the patient of your atherosclerosis. Alright, so this is all about in your lipid metabolism. Now next comes our protein chemistry. In protein chemistry, important is classification of amino acid. Important for the Viva. In amino acid classification, you should know first what are the essential amino acids and why it is called essential. So you should know the name of all essential fatty acids. Second, hydroxyl group containing amino acid, sulfur containing amino acid. Okay. Next is that of your which is the 21st amino acid and which is the 22nd amino acid. What is the name of 21st amino acid? What is the name of 22nd amino acid? Next is that of which is the ketogenic amino acid, which is both ketogenic as well as your this thing glucogenic. You should know the name of which amino acid is ketogenic, which amino acid is glucogenic, and which amino acid act as a both ketogenic as well as glucogenic. Important name of your aromatic amino acid. What are the examples of your aromatic amino acid? Next, which amino acid consists of imino group? IMINO. Amino group. The answer is your proline. Proline is the only amino acid which consists your imino group. You should know the which amino acid consists the imino group. Okay. Next is your that of branch chain amino acid. Okay. You should know the name of branch chain amino acid. Okay. This is all about amino acid classification. On structural organization of protein, you should know what is the structural organization, at least name, primary, secondary, tertiary, quaternary, and what are the bonds which are present in these structural organization, and what are the covalent bond, what are the example of your non-covalent bonded protein structural organization. Examples of your super secondary structure. Okay, what are the examples of your super secondary structure that you have to know. Okay, next comes on the protein denaturation. You should know what is protein denaturation. When protein is denatured, then what bond will break and what bond will retain, and what is the principle of your heat coagulation test. Okay, so principle of heat coagulation test, which bond break in protein when you will do heat coagulation, then protein is denatured. So during denaturation, which bond break and which structural organization will break and which will retain, that you should know in the protein denaturation. Okay. Next important thing in protein comes on the plasma protein. Okay, so in plasma protein, important thing plasma protein, which is the most abundant plasma protein. Answer is your albumin. And what are the functions of albumin in the body? That you should know, and how, at least briefly, you should know how albumin maintain the plasma oncotic pressure. What is the mechanism by which albumin maintains the plasma oncotic pressure? Next, you should know the normal value of your total protein, your albumin, your globulin, and your fibrinogen. So you should know the normal range of all these things inside the body. Okay. Next important is that of electrophoretic pattern. You should know what is the normal electrophoretic pattern and what are the abnormal electrophoretic pattern in different diseases like in your nephrotic syndrome, in cirrhosis of liver, and your multiple myeloma. Okay. Important thing, you should know the basic of multiple myeloma. What is multiple myeloma? Okay, what will occur in case of multiple myeloma disease? What are the clinical features which are associated with the multiple myeloma? For that, I already shared a mnemonic of crab relating to your multiple myeloma. So you must watch that mnemonic of that video. Okay. So you should know multiple myeloma, what are the clinical features of multiple myeloma, and what is Bain-Jones protein. Very, very important. What is Bain-Jones protein? In which disorder Bain-Jones protein will find, and what is the characteristic feature of Bain-Jones protein while you will heat it and while you will cool. So you should know all about this Bain-Jones protein. Okay. Next comes on your immunology. So for UG level immunology, important thing is that of immunoglobulin. What is the structure of immunoglobulin? How many chains the immunoglobulin have? So immunoglobulin is a Y-shaped structure having 2 heavy chain, 2 light chain. That basic you should know. Besides that, you should know all the name of types of immunoglobulin. Okay, GAMDE. You should know the name of all immunoglobulin. Besides, which is the most abundant immunoglobulin out of these? Which immunoglobulin can cross the placenta? Next question, which immunoglobulin comes first in acute response or acute infection, and which immunoglobulin comes first in chronic infection? Next question, which immunoglobulin help in complement fixation? That you should know. Next, which immunoglobulin help in the opsonization? Okay. Next, out of these, which immunoglobulin form pentamer out of all immunoglobulins? And which immunoglobulin is present in the secretions? Okay. Which is the if a newly-born baby is that colostrum of milk is there, that colostrum is rich in which immunoglobulin? So you should know the which immunoglobulin are present in the secretions of body. Next important, which immunoglobulin structure exists in a dimeric form are attached with the J chain. So these are the important things that you should know in your immunoglobulins. Another important thing in immunology is you should know difference between innate immunity and acquired immunity. Difference between passive immunity and active immunity. Difference between MHC class 1 and MHC class 2. Okay. These 3 differences you must know in your immunology for your grand viva. Next important thing, you should know at least 2 examples of all hypersensitivity reaction. Type 1, type 2, type 3, type 4. All type 4, 1 to 4 hypersensitivity reaction, you should know all these with 2 at least example of all hypersensitivity reactions. That what disorder comes under type 1, type 2, type 3 and type 4. Okay. Alright. So this is all about your immunology in grand viva for UG student. Okay. Now comes important thing in protein also, that is tripeptide. Write one example or just define the example of your tripeptide. So answer is you must generally give glutathione. So you should know the composition of glutathione. Okay. The glutathione is made up of how many amino acids? It is made up of 3, and what are that 3 different amino acids are there. Okay. So this is all about your protein. Now comes on the metabolism of protein. In protein metabolism, important for grand viva, that is what is transamination, what is deamination reaction, okay, and how ammonia is formed. Okay. Next important question, if ammonia is not disposed of through the body, then why ammonia is toxic in nature and why that ammonia causes hepatic encephalopathy? Very, very important for that. Okay. Already I uploaded this video that why ammonia is toxic in nature, so you must watch that. Very important question for the grand viva. Next comes on the urea cycle. The important thing is that the site where this urea cycle occurs in the body. Okay. And why only urea cycle occur in liver only? What is the reason behind that? Okay. Next, what is the chemical structure of urea? So chemical structure of urea is NH2CONH2. So out of this NH2CONH2, what is the sources of these 2 nitrogen and carbon in the urea structure? That you have to know. Next important thing you have to know, that is the difference between CPS1 enzyme and CPS2 enzyme. Okay. The difference between CPS1 and CPS2. Next important is which is act as a allosteric activator of your CPS1. That you should know. You should know the name of all urea cycle disorder. 5 enzymes are there associated with 5 disorders. So you should know name. Important is that of what is triple H syndrome. HHH syndrome. First thing, triple H syndrome, you should know the full form of triple H. What HHH stands for in triple H syndrome. Okay. And why? What disorder will occur in triple H syndrome? Next important in urea cycle, what is the energetics of this urea cycle? Okay. So this is all about your urea cycle. Now comes on your individual amino acid metabolism. First comes your phenylalanine. Okay. First question examiner asks, phenylalanine is what kind of amino acid? Whether it's essential or non-essential. And what about tyrosine? Tyrosine is essential or non-essential, and why? If tyrosine is non-essential, then why it is non-essential? We should know the reason. Second, in phenylalanine, important thing is that of you should know the disorder of phenylalanine metabolism, that is phenylketonuria. So what is classical phenylketonuria? Which enzyme is deficient in classical phenylketonuria? What are the clinical features of phenylketonuria? Next important is why there is a mousy or musty odor in phenylketonuria patient. What is the reason behind that? Next important, what is the reason of neurological manifestation in patient of phenylketonuria? You should know the reason. You should know the biochemical basis of that. Okay. Next important thing is that although phenylalanine doesn't contain any keto group, then why the disorder is known as ketonuria? What is the reason behind that? So you should know the reason of that. Okay. Next important thing is that you should know what is Guthrie test. First thing you should know how you will diagnose the patient of phenylketonuria, and Guthrie test you know in detail. So what is Guthrie test? Okay, which agar plate you will do, what bacteria you will know. So you will know each and everything about Guthrie test. And next important is which confirmatory test you will do for the diagnosis of your phenylketonuria patient. Okay, so this is phenyl. Now comes on the tyrosine metabolism. Tyrosine, important thing is what are the specialized product of tyrosine metabolism. You should know the name of all specialized products. Okay. Next important thing you should know that how dopamine, epinephrine or norepinephrine, that from tyrosine, how dopamine, norepinephrine and epinephrine will synthesize. So you should know all the steps starting from tyrosine up to formation of epinephrine, and all the enzymes you should know how it is formed. What is difference between epinephrine and norepinephrine, and what is the role of epinephrine and norepinephrine in the body, that you should know. Now in tyrosine disorder, you should know the enzymes which are deficient in tyrosinemia type 1, tyrosinemia type 2 and neonatal tyrosinemia. Okay, this is all an important thing in which disorder you will find the cabbage-like odor. So you should know where you will get the cabbage-like odor in the urine, in which disorder. Okay. Next thing comes on the alkaptonuria. Alkaptonuria, which enzyme is deficient in alkaptonuric patient? Why there is a urine on standing black in color? What is the reason or biochemical basis of the black coloring of urine on standing in alkaptonuric patient? What is ochronosis? Okay. Why this ochronosis will occur in patient of alkaptonuria? Okay, so this thing you should know for alkaptonuria. Next comes on your tryptophan metabolism. So in tryptophan metabolism, you should know what are the two pathways that tryptophan will do. That is kynurenine pathway and serotonin pathway. So from kynurenine pathway, what is the end product which is formed from the kynurenine pathway? And from serotonin pathway, you should know how serotonin will form from tryptophan. So you should know starting from tryptophan to synthesis of serotonin, all the steps along with the name of enzyme. Okay. From tryptophan to how serotonin will form, all the steps along with their enzymes. Okay. Next important thing is that examiner asks that niacin. Okay. Niacin is a vitamin, and with this niacin is synthesized from which amino acid? So you should know through the kynurenine pathway, this niacin is formed from your tryptophan. Okay. Alright. This is next important. You should know what is Hartnup disease, what is carcinoid syndrome. Okay. Why the patient of Hartnup disease and carcinoid syndrome patient present with the pellagra-like feature? You should know the reason behind that. Hartnup and your carcinoid syndrome. Okay. Now comes on your glycine metabolism. In glycine, you should know the specialized product of glycine. What are special products formed from the glycine? And examiner asks which is the simplest amino acid. So glycine is your simplest amino acid. Okay. Now comes on your sulfur containing amino acid. So you should know first thing what are names of all sulfur containing amino acid. What is difference between cysteine and cysteine? You should know difference between cysteine and cysteine. Next important is from which amino acid taurine is formed. Okay. So important question that taurine is formed from which amino acid? So you should know. Next important is cystinuria. Okay. Disorder of cysteine metabolism. So in cystinuria, what amino acid will be excreted in patient of cystinuria? So its mnemonic is that of COLA. COLA. That is cysteine, ornithine, lysine and arginine. So COLA. Okay. So COLA is excreted in the patient of your cystinuria. The important question for your Viva, that which amino acids are excreted in patient of your cystinuria. Okay. Now comes on the homocystinuria. So which enzyme is deficient in patient of classical homocystinuria? You should know the enzyme deficiency in homocystinuria patient. Okay. And why the patient of homocystinuria are more prone for development of atherosclerosis? Okay. What is the reason behind that? So you should know the reason why homocystinuria patients are more prone for the development of atherosclerosis. Alright. And next comes, you should know branch chain. First thing, you should know the name of branch chain amino acid. Okay. So what are the name of branch chain amino acid and what is maple syrup urine disease? Briefly, you should know maple syrup urine disease. Okay. So this is all about your protein and amino acid metabolism. Okay. Next important topic is your biological oxidation. Biological oxidation, important thing is electron transport chain. So electron transport chain, first thing you should know the name of all complexes of your ETC. 1, 2, 3, 4, 5. The name of all complexes. Second important thing, you should know the chemiosmotic theory in brief. What is chemiosmotic theory? Next important, the name of scientist who proposed this chemiosmotic theory. You should know the name of scientist. Okay. Another important, what are the inhibitors of your this ETC? ETC inhibitors, one of the important question for your grand Viva. Next important is the uncouplers. Okay. What are the uncouplers of ETC? What are the physiological uncouplers? What are the chemical uncouplers? So you should know the name of physiological uncouplers and what are the names of your chemical uncouplers. And besides, you should know why uncouplers are known as uncouplers. What these uncouplers will uncouple. Okay, so you should know the mechanism. What is the mechanism that these uncouplers will do? What they will uncouple. Okay. Next important question in biological oxidation is that of high energy compounds and low energy compounds. So you should know what compounds you will show. You will tell that high energy compounds and what compounds are low energy compounds, and examples of your high energy compounds as well as examples of your low energy compounds. Okay. So this is all for today's video. In next video, we will share some more remaining topics for your grand Viva. Thank you.